Broadcast is now starting. All attendees are in listen-only mode.
Hello, and welcome to today's AAPB webinar, Change Your Brain, Neurofeedback, and the New SMR Story. We are very pleased that you can join us for the next 120 minutes. My name is Andrew Van Wash-Nova, and I will be your facilitator today. Before we get started, I just have a few housekeeping items to review. If you wish to receive continuing education credits for participating in this webinar, you are required to register for the webinar. At the conclusion of today's presentation, you will receive an email from me with the session evaluation and a 5-question multiple choice exam. You must answer these questions to qualify for CE credit. Please complete this exam within 1 week. Your CE certificate will then be emailed to you 1 week upon verification of the exam and evaluation questions. Further, all attendees are muted. Please use the question feature to send questions to me directly, and I will compile them for our presenter. We will address all questions at the conclusion of the webinar. We will conclude today's webinar at 3 p.m. Eastern Time. If we are unable to get to everyone's questions, I will send those to our speaker after we end, and all questions and responses will be emailed out to you once those have been addressed. It is now my pleasure to introduce today's presenter. Dr. Barry Sturman is faculty for the Collaborative Centers for Integrative Medicine at UCLA, as well as Professor Emeritus for the Departments of Neurobiology and Biobehavioral Psychiatry for the UCLA School of Medicine. Among his numerous accolades and achievements, Dr. Sturman was a founding member of the AAPB, has been involved with over 100 published peer-reviewed articles, has contributed to more than 30 chapters in published books, and has helped to shape and influence the field. We are very honored to have him present with us today. It's now my honor to turn it over to our presenter, Dr. Barry Sturman.
Now I go to where it says show my screen.
That's correct. Yep. Go ahead and click that button. There we go.
Okay. Good morning. I'm in California, so where I am, it's a morning. I don't know where you are. It's a great pleasure for me to have an opportunity to speak to all of you, and I really thank you for your willingness to come in and spend 2 hours listening to me pontificate. I haven't done this kind of presentation very often, but I'll get into the swing of it. This is going to be a rather long presentation because it's going to cover 60 years of my work, and it's going to end up with some very exciting new stuff. But I do get complaints every once in a while from people who've heard me speak before not to say the same things over again. And I also am told sometimes I don't speak loud enough. The problem is that this is not a talk about what I did yesterday, okay? This is a history. It's a history that builds up a consortium of facts that allows us to do some things we've never done before. And I'm very excited about that. Even though I'm retired officially, I'm still seeing clients with my students. And actually, I'd like to say this right now. I don't know. It's odd talking to a screen rather than people. I don't know who you are there, but I am now in the search for some new young trainees. You don't have to be young, who might be interested in learning how to handle this new program. I live in Los Angeles, actually in Beverly Hills, believe it or not. And we meet in my office here at home, which is nice. And if anybody, after hearing this, gets excited and wants to come over, let me know, because we do have work to do, and I have a couple colleagues and a couple students right now, but they're in medical school. The medical school students are difficult because they have heavy school obligations. So, but many years ago, I decided I didn't know what was happening in the field of neurofeedback, in the sense that there were so many different programs, different ideas, different names, different methods, and I just wanted to know what was going on. So I did a survey of some 20 different groups, most of which were prominent in the field. And I asked a series of standard questions that I thought were important if you were going to do this work. And I was extremely disappointed at the outcome in terms of follow-up, in terms of quality of method, etc. And as a result of that, one of the questions that I asked was, do you know what you're doing? And some say, I don't care what I am doing as long as it works. And I say, I don't care if it works if I don't know what I'm doing. And I hold to that very intensely in my thinking and in my work. I was early in my career, I was influenced by reading a lot of Pavlov's work. And one of the phrases that he used to describe how he felt was, don't become a mere recorder of facts, but try to penetrate the mystery of their origin. And that's what I've done throughout my career. I need to get to the bottom of what's going on. And of course, I never was able to, because it's very, very complicated. And since what we do is really a neuropsychology profession, we need to know the supporting physiology for what we're talking about. We're talking about the brain. We're talking about change the brain. One of my patients gave me that title. And you need to know what you're doing. If it works, that's great. But if it works and you don't know what you're doing and you're doing some kind of nonsense, it doesn't matter. So I want you to know that that's my bias. But I have come up in recent years, because of reviewing the data and putting it all together over a period of 60 years of work, doing many different things, but things that turned out to all speak to each other, which was extremely surprising to me. But then it became simple. So what I've tried to do is simplify. There's a lot of detail to neurophysiology. There's a lot of detail to behavioral science as well. Those are distractions as far as I'm concerned. What we have now is the ability to just look at the data and draw some meaningful conclusions. And that's what I'm going to do today, and show you the basis for that strategy. So don't worry about it. It's not going to be a bunch of neurophysiology. I have to give you some for you to understand where I'm coming from. I'm not used to doing a talk like this where I can't get questions immediately, but what I'm going to do, I've broken this down into chapters because it is the basis of a book I'm writing, and probably the last thing I'm going to do. As a result, we can take a break after each chapter. I don't need to wait to the very end to listen to questions unless that's demanded. If there are questions when we take our breaks, I'll be happy to try and answer them for you. I don't want anybody here to leave saying they didn't learn anything, because this should be a major learning experience for you. And so I'm counting on your cooperation in that regard. And by the way, I have a system that's recording you, and I can tell if you're falling asleep, so be careful. Okay, so let's move on. Chapter 1 is called Fundamentals First. And for those of you who snore and say, Barry, oh, we've heard this before. Okay, tell me if you can answer these questions. I have some friends out there, I know that, and I'm throwing this at you too, because you're the ones that tell me I always talk the same way, and I don't. Okay, so how does the EEG inform brain changes? Write down some notes. What neurophysiological events set this process in motion? Which basic functions best reveal relevant behavioral changes? Okay. Ask yourself if you can answer all those questions effectively, because they're fundamental to what we're supposed to be doing. So we're going to start with fundamentals so that we're all at least on the same page when we move on, whether you've had or have heard them before or not. And the first topic we're going to talk about is where EEG rhythms come from. And I suppose you all appreciate that that's important. Okay, so this is a slide lots of my friends have seen before. It's from Netter. It shows a picture of the forebrain of the human central nervous system with both a lateral view and a medial view. And then the various nuclei that make up the relay systems of the thalamus and where they project to on the cortex using color code. I show that first because actually I'm going to use an analogy of a railroad system. And this is Grand Central Station here. These are the tracks coming from different stations around the nervous system, actually. They show the pathways going up. This happens to be a somatosensory pathway, but going up to the Grand Central Station, which is up here, and how they're projected up to the cortex from there. And we're going to go over that in some detail using the metaphor of the train system. All of the inputs to the cortex are of sensory origin. They are telling the cortex what the rest of the body is doing. And they all involve 3 neuron pathways. They go to the thalamus, as you see, ending up here. And they come in at different levels. Some come in posteriorly in the brain, in the spinal cord. Some come in the brainstem. And they all join up as they go up to the cortex. And on their way, they enter into connections or associations with other pathways too that join them. So it is actually kind of analogous to a railroad system. You have trains coming in from different stations, going to Grand Central Station, and then when they get there, it looks like this. So here's what we have going to the thalamus. We have ascending afferents. That means from the somatosensory system, muscles and joints, cutaneous sensations, touch, pressure, pain, temperature. You have brainstem afferents, which are the same functions but coming from the brainstem level. And we have, believe it or not, and I was even surprised to appreciate that, basoforebrain afferents. Now, the basoforebrain is going to be an important part of what we're talking about today. And so I am glad that they had that on here because this isn't my slide. I got this out of a paper I was reading. All of these pathways go to specific nuclei in the thalamus. Those nuclei are not really color-coded, send their axons all to layer 4 of the cortex, which is the sensory afferent termination. From layer 4, they go up to the cortical surface. We'll talk about that later. Ascending afferents again from the eyes, from the ears, from the somatosensory system, I said from the cerebellum, from the basal ganglia. All of that is sensory stuff going into the brain. And on the way up to the cortex, it gives off a collateral pathway to another thalamic nucleus called the thalamic reticular nucleus, or the nucleus reticularis. They always have many names for these things. And their role we'll discuss in just a moment. So here comes the train, comes to the station. It goes to it's switched to the right track that takes it where it's supposed to go, and it's heading up to the sensory pathway. And in the sensory pathway that goes to layer 4, but for the system it's related to. So for example, for the sensory motor system, you have the ventral basal nucleus, which carries in both lateral and medial pathways from the body. The medial ones are from the brainstem, the lateral ones are from the spinal cord. So there's a bunch of anatomy, and it's nice to know it. It isn't essential, but it's nice to know. So now back to the analogy. Here's the train coming up the track. It's taking passengers either to or from work. Okay, if they're going to work, they're going to have an excitatory effect on the cortical neurons they eventually connect with. And the last hub before they get to the cortex is at the deepest layer of the forebrain cortex. before they get to their destination. If they're excitatory pathways, they're going to work. So they're going up to the system, but they're going to have an effect on the cortical, thank you, on the cortical neurons in the area that is their destination that is excitatory. It activates those cells. If they're on a team that's coming home from work, they're going to go to the nucleus reticularis. They all go to the nucleus reticularis after all, but the ones here are particularly important. And they're going to feed back to the thalamic cells. Now we call these relay cells, which is an earlier name. Now some people call it first order nuclei. But these relay cells are relaying what's coming up these ascending afferents and going to their cortical representation. The lateral, pathways that are deviating from that into the reticular nucleus are actually telling these cells what the destination is like and what it needs and what it thinks is important, okay? So you may be sitting in a room, I don't know where you are listening to this, maybe there's an air conditioner on, global warming after all, or noise coming in from the window, and you don't hear that until I mention it. And then I tell you, I say, the lights are on or something. And oh yeah, the lights are on. Otherwise, because it's not relevant. If it takes on priority, it is relevant, and that relevance is fed back from the cortex through this system to the thalamic relay cells, and it changes their behavior. So it's very important. This is a very obviously, unlike a machine, or although nowadays they're pretty sophisticated, it's very flexible and it's very responsive to relevant conditions. And that happens by way of connections from the cortical level back through other, these are called pyramidal cells. They look like pyramids here. And the pyramidal cells feed back both to the reticularis and to the relay. Now, there's an issue that this reality, let me just see if there's anything else I need to share with you about this. I hope that is sort of clear. Let's stick with the train. If this is a crew coming in, going to work, this is an excitatory effect. Oh, come on, cut that out. If it's a group coming back from work, it's going to be, they're going on vacation. So it's a different effect. And we're going to get into the significance of that differentiation in other ways all the way through this talk. So hopefully you'll understand the importance of the gas end brake of the central nervous system. Now, one thing that's interesting is when these various railway tracks enter different stations or hubs, they interact with other tracks coming in from other locations. And those interactions constitute networks. Now, another thing we're going to run into that's very important and needs to be appreciated because it's relevant to what we're talking about is if there is damage to any of the connections coming from, let's say, the thalamus and going to the next way station in that sensory pathway, it can be compensated for by an alternative pathway being established, as we see here. So nothing is lost, and that system continues to operate. And that's becoming very relevant to what we're supposed to be doing. And we'll be seeing that, and I hope that you all wet your pants. It's really going to be interesting to you. That's all I can say. So keep that in mind. We're going to be talking about network interactions with this system. Now, what happens when the people that are going to work, those get to the destination they're going to, the train track changes. It starts to do something which we call depolarization. And we'll look at that in a different way in a moment. This is a cell that is polarized, and and is depolarized right here. And now it's going to polarize as this information comes in. And when it does, the cell will discharge. It's going to let an electric charge go. And that's activated. So an active cell used to have a wonderful tape of the sound, and sound is what this really does. It goes boop, boop, it goes boop, 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 boop. Now it's the electrical difference between the inside and the outside of the membrane of that cell is basically at about 30 microvolts usually when it starts to do this. That means it's being activated. It's going to have a reducing charge across it. It makes the cell easier to excite. As that cell becomes suppressed, like it's going on vacation, we see a thing which we call inhibition, and it goes down to a larger polarization across the cell, and it's starting to become what we call hyperpolarized. But it reaches a point where it's not firing at all. Okay, if that decrease in excitation, this is being caused by excitation, this response here. If that keeps decreasing, instead of just being silent, that cell begins to do something very unique. It begins to burst. And you can listen to that, and it goes brup, 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 brup. It really does sound like a train, by the way. I wish I could know who took that particular video I had. So there are two kinds of inputs to the receptive cell from the train coming into that station. One is excitatory, so it's called an excitatory postsynaptic potential. What it's doing, here's the threshold determining nature part of the cell membrane, and if it's excitatory, it's moving positive ions into that region and becoming more positive outside, negative inside, and therefore it's more highly polarized, or depolarized, I'm sorry, negative inside, positive outside. So it doesn't take too much excitation to fire it. And the more depolarized it gets, the more receptive it is to activation. On the other hand, you'll see there are certain chemical transmitters that produce that. On the other hand, there are inhibitory neurotransmitters that cause the opposite effect, where the positive ions remain outside, and this carries more negative ions inside. So now there's a greater difference between the outside and the inside in terms of positive and negative ions. And of course, when the cell gets activated, they come together, and the negative ions raise the threshold for activating that cell. So a cell that's been stimulated by a neurotransmitter that causes a inhibitory postsynaptic potential. I'm having trouble with my mouse, by the way. Is harder to activate. It takes more current to depolarize that cell. So that cell is relatively, to various degrees, depending on its inputs, inhibited. When the cells reach that, go back here to this one, to the point where there's sufficient hyperpolarization. This is hyperpolarization. The hyperpolarization occurs when these inhibitory inputs reach the membrane. And when that happens, if it reaches a certain level, as we just saw, those cells will start to burst instead of just firing. And that bursting will come in to the nucleus reticularis, that collateral coming off the pathway to the cortex, and cause those cells to burst. And when those cells burst, they give off GABA, which will go back directly to the cells that they came from, that the input came from, and cause those cells to be depolarized. Okay? And that depolarization leads again to hyperpolarization. And you get this oscillation between the reticular cells and the relay cells in terms of pattern of firing. And it's this oscillation that is the basis of the EEG. That signal is projected up to the depth of the cortex, your area 4, as you know, and it's negative in the depth and positive at the surface. So you have a current flowing there, and that produces waves in the EEG. Now you take a better look at that here. This is actually a recording we made in my lab showing, we're jumping ahead, this is SMR, showing a 12 to 14 hertz filter being activated by a BPL cell. That's a ventral posterior lateral nucleus cell coming from the thalamus and going into the bursting mode. The bursting mode is producing the frequency that is seen here as we pass the signal through a frequency analysis, and that's associated with the actual rhythmic pattern in the EEG. That's, of course, how we learned what the frequency was and that the cell was being activated by this bursting activity. Well, whoops. Let me just go on to this other thing first, because we're going to come back to this in a second. So this is the model, rhythmic self-firing circuitry. And I first saw this slide and the subsequent slide at a meeting in Florida that we had when SABA was still active. And on that, the grounds where that meeting was held, the University of Florida Laboratory of Marine Research was on the same grounds, and some of their professors came and spoke with us. And it turned out that this is the same kind of rhythmic circuit. This would be a circuit. Let's say the thalamus, this is the reticularis, interacting and causing this rhythmic pattern and continually showing it under certain circumstances. I thought, well, that's exactly what happens in the human thalamus. However, this came from the stomatogastric ganglion in the lobster. And this is a system that causes peristalsis in the digestive tract of the lobster and moves whatever the digestive tract is getting rid of down pathways. So there are many instances where particular systems of operation in the nervous system have been around for eons and are borrowed when they are useful in other situations. The thalamus came late in the evolution. These things came a lot earlier, I'm sure. And so it's nothing new under the sun. It's really very interesting to recognize that evolution is a progressive process that doesn't forget what it's learned. Kind of amazing to think of it that way. I thought it was worth taking a detour and showing you that. So they're saying it's one of the premier model systems to study network dynamics in the nervous system. All right, so now we got another problem. A lot of you know of a method called Loretta, right? And unfortunately, some people have labeled that as source localization. Well, you should be able to know from what we've just learned that that's not true. The source of the signal that's going to the cortex is coming from the thalamus. Now, if you happen to know what system you're dealing with, and you know that it's affecting that system, you can say, yeah, it's coming from the eyes or something. But you don't always know that. In our situation, we know that. So anyway, a better term for this is not source localization, it's source target, where it's headed. It will tell you that. But it's unfortunate because there are limitations to it, and people are using it a lot, and nobody seems to ever mention that. I have read some papers where they've mentioned that. So this is a preparation that was created and done by a colleague of mine at UCLA, Jaime Villablanca was his name. He doesn't play baseball. And Jaime removed the thalamus in a series of cats and studied their behavior. And he labeled them the athalamic cat. So again, those of you who know me know what's coming up next. I wrote a poem about the athalamic cat, just so I wouldn't forget what it was standing for. And I'll try and share that with you if I can get my voice back. This is the athalamic. This is not the pussy in the hat. This is the athalamic cat. He can't make alpha all the day. He can't make theta anyway. It's delta delta all day long because you see his thalamus is gone. No rhythmic activity can come out of this brain because the mechanism necessary for the rhythmic process is gone. And so there goes your source localization nonsense. Okay, then in addition to that, the studies out of Yale, where instead of removing the whole thalamus, they severed the connections between the relay nucleus, this happens to be the lateral geniculate nucleus for visual pathway, and the nucleus reticularis, that's called the parageniculate nucleus here. And in the region where that connection has been severed, there is no rhythmic activity. Okay, above that and below that, it's normal. So there's no question that rhythmic activity is created in the thalamus by virtue of the process we discussed. And it's important to know that when you see rhythmic activity in the EEG, no matter where, under normal circumstances, it's coming from that system turning down, being inhibited, being suppressed, okay? It confuses a lot of people because more rhythm means less function and vice versa. Keep that in mind and just think of the athalamic cat if that helps. Now there are exceptions to that. For example, if there's damage to the tissue, you can have abnormal high frequency activity from tissue damage, and it has a rhythmic-like component to it, but it's very fast, and you'll see some. Or it might be a 6 or 7 year old child who's just closed his eyes and is putting out a frequency different from what you would see from that a certain part of the brain, and they call that pediatric alpha, although if you look at it, it's actually theta. It's a slower frequency, but it acts like it's alpha in the adult. So there are some problems with the language that we use, and it's useful to know that. If you're talking about an epileptic or sometimes a head injured patient, you should be careful what you label what you see in the EEG. And if you're talking about an adolescent or a child, you won't really see normal eye closure alpha in a human until they're between 8 and 10 years of age. Prior to that, it's a slower frequency. Now, it turns out, did I get this one? No. Oh gosh, it's an important slide that has somehow slipped between the cracks. Ah, here it is. Okay. So I just showed you some data, I think that was this one, of our recording of rhythmic activity in the 12 to 14 hertz range over the sensory motor cortex from a activation or burst production in a ventral basal cell. This is from a study by Hughes and Crunelli in 2007, showed that exactly the same thing. Here's the sensory motor rhythm. Here they got different recording speeds. And it's coming from VPM, ventral basal nucleus, starts to burst, and here's the alpha wave, which originally, unfortunately, Berger is the one who named this the well, he didn't name this one, he named the alpha waves up here. There's another European neurologist who decided to call this activity mu. And you'll still hear mu in the literature or in some people's talks. This is a man that I knew, and he was somewhat strange, and I think he hung out with cows too much, as far as I can figure out. Anyway, this is SMR here. Then there's theta, and there's alpha. And you look at this again, all of these important sites, important EEG patterns. Alpha occurs when the eyes are closed. What happens when the eyes are closed? Excitation is reduced, okay? You're turning off the source of stimulation for that visual pathway. In addition to that, without any input to the system, the cortex can't evaluate it. So you're losing excitatory input from the cortex too, to that visual input. The theta waves, as we'll see later, are coming from a different part of the brain, but they come in through the thalamus just in the same way. And here you can see that when we blow up the pattern, you see the bursting in the cells and the development of rhythmic activity again, okay, in all of these situations. So we know what makes the rhythms we look at, okay? It's not a mystery anymore. And we know, we certainly should know where they're coming from because they're landing in the place of that where that pathway ends. And we're going to get into more detailed discussion of this later. I guess I hit the wrong button. Let's go back to get caught up again. So you remember the athalamic cat, don't forget it. And this is, at one point, this is back in the early 70s, I think it was, that we published a paper with Bowersox and myself, showing actual thalamic recordings along a pathway that we felt was responsible for the SMR, which we'll get into. That's what this is going to be about. And we're recording various things. On the afferent side, afferent means it's signals going into the cortex. If the individual becomes silent, this is called a somatic afferent. If you're stretching the muscle or stressing the tendon, that pathway will fire. It tells you about the status of muscles. And so if it's not being activated, the cells shut down. They stop firing. That pathway goes up to the thalamus. Again, this is Grand Central Station. And on the way up to cortex, we're going to go to the motor cortex. It throws off, it goes into the nucleus reticularis, causes bursting there, bursting in VPL, and that is then conveyed up. You should see the connection here, but it's conveyed up from the thalamus to the sensory motor cortex, and you get the sensory motor effect. Now there's an interesting thing right here. What we did the first thing we did when we discovered the sensory motor rhythm was try to figure out what it was connected to. If there was any other changes in EEG or electrical activity corresponding to or correlated with the SMR. And one of the first things we saw, which was very profound, I mean, we looked at the source of the spindling, which was the rhythmic reciprocal activity between the reticularis and the thalamus, thalamic relay. There was a clear pathway directly to a structure called the red nucleus, the RN there. And when someone was producing trained SMR, they were shutting down that pathway. It was being inhibited, okay? Which has created a puzzle for all of us recently, because there's a wonderful laboratory in the Netherlands run by Ingrid Philippins, who's been using SMR to look at Parkinson's disease. She was led in that direction by a very small paper written by Michael Thompson when he ran a friend of his in their program. who had Parkinson's and gave him some SMR training and saw that he got better. And he wrote up a little paper on it, and thank God he did. I don't know if Michael and Linda are listening, but if they are, thank you. I've said that to them many times. So question is, her interpretation of why, well, let me back up. She finds that if she trains, she uses an advanced primate model, a monkey, a vervet monkey. There's a drug that causes the onset of Parkinson-like symptoms in the animals. And so she's got a model for Parkinson's. And she looks at drug effects and various other things. And so she had seen Michael's paper and she knew them and tried SMR training with some of these monkeys that were showing Parkinson-like symptoms. And sure enough, the symptoms fell off. The animals got more normal. And according to her, the red nucleus got bigger. She studied the size of the red nucleus. And she's interpreted that as the excitation from cortex that the SMR produces stimulates the growth in that particular nucleus. And we need to talk about it some more because actually the nervous system is a very plastic thing. And lack of input can either, let me back off again on that because we'll come to that. What the system that produces SMR does primarily is inhibit. It's an inhibitory system, and we'll come to that. And so we assume that, and we don't have to assume because there's the cellular pattern. These are real recordings of the cells showing an inhibition during the SMR. So that nucleus is being inhibited by the SMR. So it's curious, what is the inhibition making it smaller? Because she says in her papers, in her studies with these animals, that the animals who develop the Parkinson symptoms get larger red nuclei. So maybe it's normalizing that, but the interpretation of it is something I completely agree with, because it turns out that the red nucleus, which you see here, is right next to the substantia nigra. And it's loss of the small cells in the substantia nigra, or damage, that elicits Parkinson's disease, or leads to Parkinson's disease. So it's very possible that cells in the red nucleus are an alternative pathway for achieving stability in that particular system. And that takes much, and I'm going to be able to show you some more data, but I can't wait to get Dr. Philippines into California so I can talk to her. I hope someday we'll get a chance to talk to each other. And I will be talking more about her work, which is very good. Okay, so that's the end of chapter 1. And we're going to start now on talking directly about clinical effects and the underlying process for that. I think probably some of you wouldn't mind having a few minutes off right now. And I hope you can hear me. If you can't hear me well, please let somebody know.
Dr. Sturman, if we're going to take a break, we did get a question come in during your, probably about 6 minutes ago,
Okay.
that I can kind of read off here. Have you also considered the role of 40 hertz thalamocortical, sorry about that, event binding rhythm, which originates from the medial nuclei of the thalamus, scans the cortex, and returns the information to the central thalamus, where it is integrated. This event binding rhythm was discovered through, I'm probably not going to say this correctly, but magnetoencephalography. Encephalography. Sorry.
I know the guy, or I knew the guy before he died, who first started that work.
Okay. And they go on to say, I think this is really the primary system involved in understanding. We can distinguish this from the other 40 hertz via an algorithm.
Well, sure, and that there's literature on that. It's been somewhat controversial from time to time. I'm sure he would admit that, because it's so close to the normal arousal pattern, frequency in the brain, the abnormal high frequency stuff, which you can get from lesions, you can get it from anesthetics, all kinds of things. So it's always been tough to record, and it's a movement artifact problem too. But I like as I said, and unfortunately it's been so many years, I've forgotten his name, but he was a nice fellow, and I think he did very nice work in describing that. So I don't know, but from our perspective, as you'll see, we've kind of focused. I told you, parsimony is the name of the game for me these days. Okay, there's 8 million other things going on all the time, and you can talk to 30 people and get 30 different ideas or explanations. I don't make any theoretical conclusions. That doesn't sound right at all. Of course I do. Most of my thinking is based on fact. It's based on data that I've seen with my own two eyes, that I've produced in my own program or with colleagues that I know well. And so I just stick with what I can trust. There is a lot of, I have to be honest with you, there's a lot of misleading stuff in the literature. I've mentioned a few things about it already. So, and I'm not saying the 40 hertz stuff is misleading. It's just, it hasn't been, to my knowledge, I've stopped following it years ago, but maybe there's more data on it now than there used to be. But that doesn't mean I'm putting it down. I'm just saying it's a hard thing to study. So in response to the question, which is a good one, perhaps if you see the method that we've described later tonight, maybe it will be tonight, maybe you can put the same information to a different kind of test. One of the things that I'm finding out, I know I'm one of the guys who started QEEG in this field, and and I still use it when it's appropriate. But I think what we're doing now gives us an alternative to that and to a lot of other things. So let's wait until we get to that, and then I can talk to that gentleman. I'll be happy to. So can we take a 5-minute break? Andrew?
Sure. Yep.
Okay, so let's do that. Stretch, move your body around a little bit, which is very important, I'm finding out. And we'll start up in 4 or 5 minutes. And this will be chapter 2, which I thought was very fundamental and important to me. Okay. If you have any other questions, jot them down and hand them to Andrew, and I'll do my best to answer them. I'm upset with myself for not remembering that fellow's name, but I do remember him very well. Anyway, have a nice break. We'll start up again. By the way, if you're taking a break, some of you may be sitting back in your chair and closing your eyes. What do you think your EEG is showing? Of course. Okay, and where is it coming from? Of course. Okay, those aren't difficult questions. Okay, maybe we don't need as much time as I thought. Let's get started again. Chapter 2. I must say that I find it difficult to talk to a screen. It's not anything I'm used to or like. I like direct contact. And the other thing I want to point out is, unfortunately, I won't be able to turn this particular work over to you for your own copying because it's going to be a part of a book. And I have to go through the technical process with that of making sure that it's original. But I will, anybody who's taken this course and wants to get that book when it's out, and I think AAPB is going to be the publisher, and they'll know where it is if it's not coming from there. I will arrange for a meaningful discount, okay? Sorry about that, it's just something I'm stuck with. Okay, everybody ready to go back to work? Okay, I call this a fundamental epiphany in the history of neurofeedback and EEG and autonomic correlates of cold pressure stimulation. When I was an undergraduate at UCLA, I saw in my junior high school manual that when we graduated, each student was asked what they wanted to do when they grew up. And honest to God, when I looked at the picture of me and what was written under it, I wanted to be a scientist, okay? And apparently that was true because as an undergraduate, I published 2 papers and got off the ground. And because of that, I was awarded a grant from the federal government in the VA system. The VA system was pretty much changing at that time, and they wanted to add research as a part of what they had to offer so they could recruit better medical staff. And at that point is where I came on the scene, and they offered me a grant to work in the VA there. And I ended up working with a professor from UCLA, who I had taken a course from, named Marion Wenger. He's written a book on physiological psychology, etc. Given a name like Marion, he changed it to Gus. So we all knew him very well. He was a nice guy. This was shortly after World War II, and he and some colleagues had come together with the notion that if you could describe the pattern of autonomic response to various pleasant and unpleasant experiences, you could codify those experiences in terms of their effects on the body. And that was an important part of his research, and he became famous for it too. And at the time that I joined his laboratory, he had gotten a laboratory at the VA hospital at Sepulveda in Los Angeles. And I went to work in that particular laboratory, recording autonomic correlates of a very unpleasant stimulus called cold pressure stimulation, where a subject put their foot in a container of -4 degrees water, ice water, and had to keep it there for a period of time. At the time, there were other graduate students in now I'm in graduate school, other students in that lab, and they were autonomically oriented. And autonomic data are collected by recording slowly on polygraphs because it's slow stuff. I was already an EEG freak, and I decided it would be really interesting to know what the correlates would be of the autonomic effects on the EEG. And that meant recording fast. And so we had a little bit of a dispute about that. And I went to Wenger and I said, look, we can do that now. Could we please add the EEG to the protocol? And he agreed, and we did. And I learned then that something my sister had taught me: if you never ask, the answer is always no. So it was an important experience. So we added the EEG, and we ran 100 neuropsychiatric patients at the Sepulveda VA hospital through this protocol. And again, because of time, I'm going to race through this. I have to. The protocol was the subjects lay on a gurney for 15 minutes with a shoe and stocking off 1 foot and a bucket of ice cold water right next to the gurney with a light attached to it. The light was associated with a pedal at the bottom of the water containing, I don't know what to call it, pot. It was a small, kind of like a lunchbox that had water in it. They had to push the pedal down when they put their foot in, and that turned a red light on. And that red light had to stay on for at least a minute before they could pull their foot out. They were then given instructions to put their foot into the ice water. They began the cold pressor stimulation, and for 1 minute they kept their foot in the bucket, and after that they had a 15-minute recovery. So we did that with all of these patients, and we recorded EEG and many other things. And what we saw, we'll start with the EEG, we found 3 groups of subjects among that group that we actually recorded from. And one was a group that was constantly active. They had some alpha in the pre-stimulation period, but not much. And they began the cold pressor and became activated and remained activated for the next 15 minutes, which is the only period that we recorded. The recording was actually bilateral, parietal, and some in the somatosensory area up here. And what we saw was there was another group that had a rapid alpha recovery. They had a fair amount of alpha in the baseline period before the stimulation. And as you'll see in a minute, during the cold pressor, they activated with the instruction and they putting their foot in the bucket. And as soon as it was in there, they went back to alpha. And they had sustained alpha during the recovery. So that was interesting. So what we actually came up with, well, here's some of the autonomic data. Respiration, the prolonged alpha block group held their breath in the cold pressor situation, and then afterwards became very unstable in their breathing for the whole recovery period. The recovery group, the rapid recovery group, increased their rate of respiration during the cold pressor, and really even during the cold pressor stimulation began to recover to the baseline level. Let's look at that a little longer. And again, they came down and returned to normal, even lower than they had been in the baseline. And then the third group, oh no, this is still the 2 groups. So we have diastolic blood pressure here. They both start out in the same place. The cold pressor stimulation caused their diastolic pressure to go up in both groups. But during the cold pressor, both of them showed some change, but the rapid recovery group showed a very rapid, I have an uncontrollable right arm, recovery, and again, returned to normal, even below normal pressure in the recovery period. So we actually identified 3 groups. One was stimulus bound, that is, they put their foot in the bucket of ice water, they activated, and as soon as they took their foot out, they go back to normal. So those were, we call those normal subjects. The second group was a prolonged arousal group, about 25% of the patients. They put their foot in the bucket of water and never recovered from the arousal. Well, at least in 15 minutes. And I consider that to be anxious people, and I'm more and more convinced of that. And the third group was a rapid recovery group, again, 26% of the patients. And I interpreted what they showed as escape from an unpleasant situation. which suggested something that I had already come to conclude from my other work, that must exist. Because you have to remember that at this time, when people talked about the nervous system, they talked about arousal. It was the period of the brainstem reticular formation, which, by the way, was discovered by H.W. Magoon, who was head of the Brain Research Institute at UCLA, and someone that I knew. And again, I should indicate that by this time I was closely associated with that program with the Brain Research Institute. And it was a hotbed of neuroscience. It was really every famous neuroscientist in the world came through and visited or gave a lecture or whatever. So it was a great place to be, and still is, as a matter of fact. So we had these three groups, and the rapid recovery group is the one that most interested me, because unlike the arousal and even the recovery response, the normal response, it indicated that there was some mechanism in the central nervous system that could, if you wish, protect a person from discomfort, give them a way to get away from it without suffering too much. So that was not a popular idea. The idea was for sleep in particular, which I was also interested in, that sleep was the absence of arousal. So when the reticular formation turned off, that's when we went to sleep. Well, I didn't make any sense to me, because I got to look at my watch and say, I have to go to bed because I have to get up early tomorrow. And it had nothing to do with the lack of arousal. So I wanted to solve that issue. Okay. And then I became familiar with Pavlov's work, where he was doing various things with dogs in slings, doing classical conditioning. I think we all know about that, what that's called. But he did one kind of thing he did was to train dogs to salivate, sugar paste or something put into their mouth, and associated that with a particular tone. And when he sounded that tone, they would salivate, because they knew that meat powder was coming. And once they were well trained to do that and salivating with the tone, he then began using a different tone to give a shock to the hind leg. And a different response, of course, was withdrawal of the hind leg and being unhappy. And when they conditioned that, both the salivation from the higher frequency tone, and the tone related to the shock stopped having their usual effect. So here's the dogs. This is a younger dog, but basically go to sleep. I like to think that they were thinking, the pox on your experiment, I'm out of here. And so he saw this sedation response, which he named internal inhibition. And I thought that was great. I got very interested in that. So we decided to follow his instructions, accumulate the facts, and start to penetrate the mystery. So that's chapter 3. There were earlier studies in the literature that I had found that showed that electrical stimulation of an area at the base of the forebrain in cats produced some of the same behavioral and autonomic changes as seen with internal inhibition. But there was no comprehensive EEG data available at the time, and I was driven to fill this gap. Okay. There was something I wanted to say, but I've forgotten about it. I was very fortunate, because at the time I was taking medical school courses, and I was taking a medical anatomy course, and one of the professors was Dr. Carmen Clementi. And I thought he was a good lecturer, a young man. I was from Pennsylvania. He'd only been there for less than a year. And I ran into him in the lunchroom at the Sepulveda VA hospital and asked if I could sit down and talk to him, because I had a question I wanted to ask, or I had an idea I wanted to share with him. And he was very accepting. He said, sure, sit down. I'm bored. So we sat down together and we talked, and I told him the story that I thought it would be very interesting to see what the EEG correlates were of the autonomic arousal produced by a cold pressor in human subjects, right? Because we had that at that time, and was stimulating that area going to produce any kind of EEG effect. And he thought it was a good idea. And we ended up, he became my professor. I worked with him for a long time. He became the chairman of the anatomy department and of the Brain Research Institute at UCLA. He was also the best man at my wedding, and we had a great fun making science from 1962 to 1974. He passed away a couple of years ago, but he was more than a brother to me. So we began exploring that area called the basal forebrain. And here's what it looks like in a cat. It's down the base of the forebrain. This is the olfactory bulb. Hippocampus is here. This is in the human brain. And the connections from that basal forebrain area go to the thalamus, they go to the brain stem, they go to the spinal cord, and into the limbic system too, and around various areas of the cortex. So it is a very profoundly networked site. And the first time we stimulated it, by the way, I passed out when we were, or I didn't pass out, but I almost passed out when we prepared this animal. We were doing acute preparations so we could paralyze them and put them in a stereotaxic instrument and see what happened when we stimulated their brain. Can't do that sort of thing anymore, but it was very useful at the time. And we just turned the stimulator on the first time at a very low frequency and amplitude. And sure enough, here came this wonderful rhythmic activity with a burst-like quality to it. And it was sustained as long as we stimulated, and the animal would fall asleep if he was not aroused when we started. And then we would wake him up and stimulate him again. So in the long haul, we produce these nice spindle bursts that look very much like what we call stage 2 sleep or non-REM sleep in the cat. Now, for those of you who think animal work might not mean much, let me just show you looking at natural sleep in humans and natural sleep in the cat. This is stage 2 sleep. These are sleep spindles being derived from a certain part of the cortex near the vertex. And in the cat, sleep spindles being derived from the sensory motor cortex, which is basically right about here in the human. So this is something that started up, I guess, before it got to human evolution and continued in the human evolution. So this is a process that shows that the cortex is being inhibited. Remember, rhythmic patterns under normal circumstances will mean the system is shutting down excitation to a certain extent. So if we have this is our first paper we published together. If we have a cat who's not doing anything in particular, we had a system developed that we could record and the cable wouldn't get tangled up. And we'd stimulate and the cat would lay down and go to bed. And we could stimulate, put him to sleep, wake him up, give him a while alert, and stimulate him again, put him to sleep. And EEG rhythmic patterns and bursting came on. On the other hand, if the animal was doing something that animals like to do, as you see here, we put a mouse in the cage and the cat went after the mouse, and we turned on the stimulator and he lost all interest in the mouse, became quiet. The mouse was relieved, I guess. And we tried to kind of describe this particular kind of behavior, this pattern of behavior. a lack of any decision as to what to do. I mean, he was just out of it. And we did a number of different studies. This is a cat who's trained to eat food at a cup. And we turn on this thing, cats, when they are offered food, are very tenacious. And we turn off the stimulator. He'd leave the cup as if he wasn't interested at all. And as soon as we turn the stimulator back on, he'd go back to the cup. So, and then we did a series of studies where we made lesions in that part of the brain. And the animals developed hypersomnia. They didn't have any meaning wakeful behavior. They weren't dying. They were okay, but they just were sleeping all the time or resting. Now, it turned out that in the early 1900s, a neurologist in Europe by the name of von Kahnemann was studying I guess it was this is what happens. A problem that had developed in Europe where a lot of people were developing two different kinds of abnormal sleep patterns. One was hypersomnia, and the other one was insomnia. And he studied the brains of people when they died from these diseases. It was a serious disease. The one that was hypersomnia, he named encephalitis lethargica, or von Kahnemann sleep disorder. Sleeping sickness. And we found that experimental lesions in the cats and the same in the basal forebrain. Oh, by the way, the lesions that he found with encephalitis lethargica were in the basal forebrain of humans. So experimental lesions in the cats and other animals produce hypersomnia, immobility, as well as a very strange motor abnormality known variously as abstinent progression. We put a cat who'd been lesioned like that in the corridor of the medical center and just let him go, and he would start walking from the medical school to the dental school, coming to a door and stopping and walking with his head against the door. And that is called abstinent progression. It's a motor abnormality, and I've never seen it before, but it was amazing. Anyway, he couldn't shut off movement. So we came to that at the time when we were doing this work. There was as I told you earlier, the reticular formation was the exciting thing going on in the nervous system, and it was about arousal. And we agreed that there had to be an active inhibitory system as well. So we thought this could be the long theorized but missing inhibitory system. We weren't the first people to suggest it. There are other people that thought this was a reasonable necessity. And sure enough, we decided to check this out. These findings suggested what we ended up doing, that it was true, that this was the inhibitory system that countered the brainstem reticular formation. And again, briefly to do that, we built this very elaborate moat with a bridge going over a water tub here. And cats trained to go back and forth between two chambers with food in each chamber. When the doors automatically opened, they could then pull this pulley, which was riding on a track, across the bridge, and along the pathway, there were photoelectric cells that recorded the speed of the animal going from one chamber to the other. After a while, in each chamber, when they had a chance to eat or drink the milk and chicken broth that we gave them, the doors would open and they could go the other way. So it was a magnificent piece of equipment, and it's hard to believe that we were able to use money from our grants to build this, and yet it turned out to be very, very effective, very useful. We also used it in work we were doing for the Air Force, which is a whole nother story. And here's what we found. When we stimulate, here was the baseline speed of this cat going from one chamber to the other, average. This is another cat. If we stimulated reticular formation they ran faster. Okay, both of them. And this we just showed two here, but that was a statistic that all the cats showed. If we stimulated the basal forebrain, they ran slower, both of them. Okay, and I still can see in my mind's eye the look on the cat's face when we stimulated basal forebrain. They'd been running down there, heading for food, and they suddenly stopped. And it's sort of like people do once in a while. Say, what am I doing in this room? It was that kind of look on their face. Then we just turn off the stimulator and they continue. So if we stimulated both the reticular formation and the basal forebrain together, they had normal baseline velocity. So we came to the conclusion that these are interacting systems, reciprocal. If the animal is too alert, this system shuts it down to some extent, or can be used just to set up another state that is more sedated, and vice versa. If there needed to be rest, they were exhausted, there was a system that could turn off arousal and let them go to sleep or rest. And that was pretty outrageous at the time. There were people who were well known in the field who questioned that, but it was very satisfactory to find out that not shortly after that, they would come back to us and say we were right. So this changed a lot of the thinking about what was going on in the sleep-waking cycle. And a lot of students came into the lab, and we got into cellular studies and things like that. And I just want to show you cellular markers of state. There was an immunostaining method called CFOS that came into use, which allowed us to map neurons that were active. And this is a paper written by one of our students at that time, in 2002. And what this stain did is if a cell was quiet, you could see the cell was able to be activated, but it didn't have much in the waking state. If that animal started to go to sleep, those cells would get very active. That's the red parts here, and they would begin to secrete GABA. You know what GABA is? It's the inhibitory transmitter substance of the nervous system, gamma-aminobutyric acid. That's the red. So this was a GABAergic system. And the studies with it showed very clearly that during waking, the firing rate of cells in that system was very low. But when animals began to fall asleep, it began to pick up in speed. And this was the rate of their firing. And as soon as it got to a certain level of excitation, we would get the reticular the basal. Boy, I'm tired. We would get to an EEG pattern that looked exactly like what happened when we stimulated the basal forebrain. And there would be motor inhibition, which then progressed into REM sleep. And we had seen many times before, because we did human sleep studies too, that in the transition from non-rapid eye movement sleep to rapid eye movement sleep, there was very frequently a burst, several bursts of this rhythmic pattern that we thought was a sleep spindle, but it actually frequency-wise was more like the basal forebrain effect. So we began to put things together, that the basal forebrain was an inhibitory system. And when you stimulated that, you inhibited motor behavior and changed not just motor activity, but the application of that activity. And we'll see that in a moment as we go forward with this kind of work. Okay, so there turns out to be a brainstem arousal system. It's in the brainstem and the posterior hypothalamus, and there are arousal. These are pathways that are identified by their transmitters. So you have serotonin, norepinephrine, acetylcholine, histamine, and hypocretin, which is a fairly recent new transmitter, excitatory transmitter. Then you have the sedentary pathways, which are the basic basal forebrain and preoptic. And I pointed out that basal forebrain to you earlier, where it's coming into the nucleus reticularis of the thalamus and then going to cortex. So, and that's GABA and galanine. Both of those are inhibitory neurotransmitters. And they interact. They go up to the thalamus. They have pathways to cortex and back. So they can activate cortical cells, and the cortical cells can activate thalamic input as well. So it's a reciprocal system. Again, requiring more detail than I can give you here, but this is basically, and then they're in competition with each other. If there's too much arousal, you get some sedentary or inhibitory effect. If there's too much sedation, you get some arousal effect, and everybody's happy. So along came another study that I encountered quite by accident. And I'd like to read this to you. I don't like reading in a presentation, but this had to be heard, and it was too much to put on a slide and ask you to read. So I'm going to read it to you. The motor cortex plays a crucial role in the generation and control of movement and motor learning. Dysfunction of the motor cortex can cause many neurological diseases such as epilepsy, Parkinson's disease, amyotrophic lateral sclerosis, Huntington's disease, and Alzheimer's. Studies have revealed that the motor cortex integrates inputs from many brain areas, such as the orbital cortex, primary somatosensory cortex, secondary somatosensory cortex. These are all things we've been talking about. Ventrolateral thalamic nuclei, VPL, VPM, the ventromedial nucleus, and in the thalamus, and basal forebrain. There are two functional subgroups in the motor cortex: primary motor cortex, which they call MOP, and secondary motor cortex, MOS, which is the secondary part, which is a little bit more towards the vertex. A study based on intracortical microstimulation revealed that stimulation of the primary motor cortex induced movements. So the movement part of the cortex was handled by the primary projection. And so they got movement of forelimbs and other structures of whiskers in mice. The secondary motor cortex is more involved in cognitive related motor control processing, such as motor decision, motor planning, motor learning, and spatial memory. Now that's a whole different category of movement. It's more than just inhibition. It works on the control and processing of motor function, motor decisions, motor play. If you had to think about those cats, the one that was looking very much stupid after stimulation and left the mouse alone, it's like he couldn't make up his mind what to do. And the same thing was true in the runway study when we stimulated those cats. So certainly motor decision was messed up in those animals. Motor planning, motor learning, and spatial memory. So it became very clear. I'm not going to use this slide. This is a slide with a axon tracking system, but it doesn't tell us anything we didn't already know. That the basal forebrain was an inhibitory system that projected to the motor cortex and that could inhibit motor excitability. Now, this is now going to be, I think, highly relevant to what we've seen in reality. So we had an idea at this point that we wanted to see what would happen if we, and this goes way back to the cold pressor studies. If we created a situation that cats didn't like, let's see what EEG pattern they would show. We found that we thought it was alpha in the humans, the prolonged or the rapid alpha recovery. It wasn't. We had primitive equipment in those times. We didn't have frequency analysis, etc. It was more like SMR, and SMR and alpha are very close in frequency anyway. So we created a situation where we trained cats to press using operant conditioning to press a lever for milk and chicken broth, which they love. And we had our cable system, which was very effective in studying moving animals. And there was a tone generator in the system here that we would then turn on a particular tone. And when that tone was on, the food delivery system shut down. So the cats would pound on the lever and nothing would happen. And so eventually they would stare at it for a while sometimes, or they would turn around and go to a corner away from the lever and just stare at the lever. They were obviously not happy. But it was really interesting that during that period, they produced this rhythm that we see right here from the coronal gyrus in the cat. And here's with the tone on. They anticipated it once we started presenting it to them. They actually could time when it was going to come. And then it got closer and closer to just having this happen when the tone was about to appear. So we actually used operant conditioning to train this EEG rhythm. And we decided to call it the sensory motor cortex because we weren't about to get into Greek and Latin, which meant nothing. This is where it was appearing on the brain of these animals. So we were able to train animals to do it, which allowed us to do a frequency analysis. And we had frequency analysis equipment, and we could tell that it was in peak frequency was 12 to 14 hertz in this situation. And then we just put them in a chamber, and if they produced that rhythm, they got the feeder operated, and we trained them to produce SMR, which is what we call it, sensory motor rhythm. And we trained the animals, and what this allowed us to do was to bring this rhythm and this process into a laboratory to study it, which was absolutely incredibly good and useful. So we could train animals and then just see what it did. So here we trained, this is a trained animal, he's producing SMR for food, and when he does so, he inhibits motor behavior. This is a neck muscle EMG, and this is integrated neck muscle discharge. So you can see that in relation to each episode of SMR, the neck muscle activity was reduced. Whoops. Come on. We looked at heart rate. Again, trained SMR activity here, looking at 12 to 14 hertz. Every time they made SMR activity, the heart rate went down. Same thing was true with respiration. I couldn't show all the things that we were using. So we saw the autonomic and EEG patterns associated with internal inhibition and with what we had seen in the humans. And of course, we were criticized. Oh, how do you know what they're doing? You know, blah, blah, blah. You're not really doing operant conditioning. So it's okay. What do we have to do to prove that it's operant conditioning? Well, extinguish it. So what you do is you withdraw the reward, and they produce more of it because that's what animals do, even humans. You take something away that they want. So we did that after here. This was 6 weeks of training and then extinction, withdrawal, and the amount of SMR increased. Another 6 weeks of training. There was more SMR during the training. We actually enhanced the production of SMR, and then with the extinction, it got much bigger. So we published this and they stopped bothering us. Okay. SMR and sleep spindles, we saw that early on. Looking at an awake animal, they produce SMR by just being still in the awake state. And when they're still, eye movement cease and motor activity is suppressed. And during the stage 2 sleep in the cat, which we call quiet sleep, we see spindles and no motor activity whatsoever. And we saw that before when we looked specifically at the motor activity. You've seen this already. There's no difference between spindles in cats and spindles in humans. That's redundant. So we did a study to see if we trained cats to produce SMR in the waking state, would they increase or alter their sleep spindle activity during sleep? And that's a complicated study, but we had two groups. One group that was trained to produce fast, low voltage fast EEG response. If they didn't have any SMR for 10 seconds, they got reinforced. Here are the reinforcements. And then another group were, I'm sorry, they weren't separate groups. We would then switch them. And this group produced SMR and got rewarded. I'm going to think about this now. A lot of this stuff was before some of you were born. And yeah, so we trained them each for about 6 weeks each in each condition and had them producing the signal. And before starting the training, we did a sleep study on them. And when we switched them, we did another sleep study, and we just followed it through. And we found very clearly that spindle burst activity in quiet sleep, which is equivalent to stage 2 sleep in humans. If the first condition, this was baseline pre, was SMR, and then followed by the low voltage fast training, the SMR increased the spindle activity in stage 2 sleep significantly. It was 0.05. And then we reversed it. We wanted to make sure there wasn't an order effect. So we did the low voltage fast first, then the SMR. And there was no significant effect with low voltage fast. But with SMR now we had 0.01 significance of increase in spindles. And that was very surprising. It's been followed up and replicated by other labs. We took the animals in the baseline after the first condition. There was a return to normal. In the baseline after the making SMR after the low voltage fast, they remained elevated, as you can see here. And then we had this contract with the Air Force, and we were looking at exposure to rocket fuel fumes and rocket fuel activity, because what they were finding, both the Americans and the Russians, was exposure to this monomethylhydrazine, which they were using in their rockets, was causing seizures and often death if they got a big enough exposure. So we checked it out in cats. So the effects of SMR training on convulsive response to monomethylhydrazine toxic rocket fuel exposure. And we'll make a long story short, the response to exposures to that drug, no matter how you give it, either as an injection or in their food or in the air, they will vomit after a certain amount of time, they'll start to vocalize, they'll pant, they'll salivate, and then they'll have a grand mal seizure. Now we had two groups of cats, a control group and the group that had the SMR training after the low voltage fast training. And what we found, and this is objective data, this is 10 cats in this group, that it took these guys, it was standard about 60 minutes for seizures to occur in the control group and 140 minutes in the SMR condition group. And a certain percentage of these animals never had a seizure. Well, that blew our mind, obviously. And it now, of course, makes a great deal of sense, and I'll show you actually even more proof of this, that the inhibitory effect on cortical neurons, on cortical motor neurons of SMR training could easily have been responsible for that. So it is important to know, and I put this in bright color so you'll remember it, in case anybody questions it in your presence. We studied, the animals we studied did not know what we were attempting to do and had no expectations. We also had no agenda outside of the scientific search. There was no issue of secondary gain or placebo effect, period. Okay, on to human studies. I'm only going to do this quickly.
Yeah, Dr. Sherman, I apologize to interrupt, but yeah, we do have about 22 minutes left for the webinar today.
You're kidding.
22.
Oh, God. Okay. Well, I'm skipping the human studies thing. These are some that most of you are. Well, one thing is, well, we'll get back to that later. This is a very important study. We had the privilege of working with some patients in neurology department at UCLA neurosurgery who were epileptics being evaluated for brain surgery. And we were doing SMR training in people who had electrodes in the limbic system and in the amygdala, and we could record what's called interictal discharge. These are pre-epileptic events. They're excitatory events in the brain. And this is just the process of training. And clearly what we found was that when we trained SMR, I hate this mouse. When SMR was present, there was no interictal discharge. So SMR suppressed the interictal discharge, and even after training, it remained suppressed for a period of time. The senior resident in neurosurgery was a guy by the name of Bloomberg, was very interested in this and worked with me on this, and we did get one little brief paper off on it, and then he was rotated to a different institute. He finished up his residency. But we did get to see this, and it's interesting that we encountered a paper shortly after that said thalamocortical neurons connected to injured epileptic cortex undergo changes and become hyperexcitable. So the neurons are hyperexcitable. How about that? In injured epileptic cortex. Inhibiting these neurons demonstrated that reducing their activity in real time was sufficient to immediately interrupt electroencephalographic and behavioral seizures. Now that's exactly what we did, because by training SMR, we were inhibiting the thalamocortical neurons. And also the changed motor neurons that also became hyperexcitable. So there was, and I wish I had more time. I'm sorry to hear that this has gone too fast. I'm not even going to talk about this. You've seen all this stuff. Some of you have. I want to get to what's important to me right now. Okay, there it is. This is supposed to be a break, but we're going to skip it. Okay, so here again we see from the work of Hughes and Brunelli and our same work how the bursting in the lateral geniculate nucleus of the visual system is associated with the production of alpha. Bursting in the limbic system and forebrain, not the basoforming, that's something else, produces theta waves. And again, based on spindling in the thalamus, and then the sensory motor rhythm here. Okay, so we have those three things. What are they telling us? The first one, parietal occipital 8 to 12 hertz alpha rhythm represents visual standby. Remember, when that rhythm is present, there is a reduction in visual processing. Sensory motor cortex 11 to 19 hertz is the SMR, and that's motor standby. And frontal 4 to 8 hertz is cognitive processing standby. So we've got these three basic conditions. And really, basically, if you think about it, if you had to pick three things to check out in terms of brain function in a patient with some kind of symptom or other, these would be some things to look at. You want to look at attention capacity, at motor behavior, and at their ability to function cognitively. So we decided to look carefully at this. So let's just look again. Eyes open, activated EEG. Eyes closed, alpha. So that's visual standby. Here's a patient who had a flap in the brain that we were able to record from, and he was squeezing a rubber ball here. And when we told him to stop squeezing it, he developed SMR over sensory motor cortex. So SMR has to do with the reduction of motor excitability, of course. Now this goes back to Ingrid Philbin's work, involvement of the red nucleus and the compensation of Parkinson's. And here's some of her trained monkeys showing she's trained SMR activity. And I had put in a description from her discussion of what they found. But what they found was a decrease in severity and incidence. I guess I can try and read this. SMR neurofeedback training indicate epochs with SMR spin on no power spectrum calculation from the, now that just describes the slide. That doesn't tell what the finding was. That's not. Okay, so what Dr. Philippins is suggesting is that the reason that these Parkinson's symptoms appear is because there's damage to the substantia nigra. That's been established in neurological research for a long time. But that the fact is there are alternative pathways in the network. And if you can activate those alternative pathways, you can restore a certain amount of the function. And the fact that the substantia nigra and the red nucleus are right next to each other is very relevant to that effect. So, as we saw before. And we see, okay, here's a math test. Every time they complete a cognitive task, they go into alpha, to theta. Oh God. I'm out of time. This is a study we do with the Air Force. These are some of my students. These are pilots that have been hooked up for study in the B-2 bomber. And we did a continuous performance test, and we could actually determine exactly what was happening because here they were presented with different letters. Let's see what's shown here. And some were targets and some were not. If they were not a target, they weren't to respond. If they were a target, they were to respond. This was the how long it took them to respond, etc. And they showed theta. Once they had determined whether it was a target or not, theta appeared. And that was very clear here. And when they were finished, they relaxed and showed alpha from the parietal occipital area. Now we call that post-reinforcement synchronization. I forgot to mention it in the sleep studies, which is a whole nother topic, but very exciting, and there are other studies going on. So I'll show you some examples of it. Okay, so this paper came out from a gal by the name of Sylvia Kober in Austria. And she showed that we could show that voluntary control over SMR activity by means of neurofeedback training led to specific improvements in the quality and intensity of stimulus processing tasks, as well as in EEG parameters, ERP, coherence, and cognitive functions. And then we went back and looked at our very first study with an epileptic, and we found that by the fourth session of training with alpha and SMR, tracking those two, at first there was no difference between them. By the fourth session, it began to separate. By the 12th session, subjects were increasing SMR and decreasing alpha. Oh, that's what happened. This is from Furtscheller. I'm not going to bother. It takes too long. This was a study we did early on with compressed spectral arrays. We trained SMR on one side of the brain and recorded from SMR to alpha. On the side where there was SMR, alpha was suppressed. On the side where there was not trained SMR, alpha was normal. So all of a sudden we realized that there were network interactions between these processes, and that became very important. And I've got to, can we really, I wanted to show this to you, but I don't think. A lot of this progress that we've made in this field has been due to technology. Lexicor started at Skill with our program, Thought Technologies Infinity, and now what we're calling the new one, just as a trial balloon, executive neurohomeostasis. I left the neuro out of that. Let's do that. This is just from skill, showing how well. This woman was hit in the head. She had very little SMR and a lot of very fast activity in that part of the cortex, frontal cortex. We trained her. SMR increased dramatically, and the fast activity in the other areas decreased significantly. So that was proof that we changed her mind. Then we came up with this system. It has a primary reward system of 4 balls, and they're each associated with an electrode site. And the subject sees this, and when he gets all of them to come to threshold, this is SMR. These are things we're trying to suppress. And again, we see alpha and SMR reciprocal. And this is left and right SMR. This is left and right alpha. You see SMR goes up, alpha comes down. We have a lot of that. Here's another. This is just looking at 2 sites. Here's SMR, here's alpha. When SMR comes, alpha goes down. SMR, alpha down. SMR, alpha down. After SMR, alpha goes up. That's post-reinforcement synchronization. SMR goes down. Here are statistics. Now, here's something you don't want to see. What do you think that represents? You're seeing SMR go up and alpha go up at the same time. Well, a better way to look at that. Let's go through it again. This time we'll use the 3 sites that we think are very important. The theta coming from the frontal area. This is F3, C3, and P3. So in this particular subject at this particular time, and we have statistics about it, they are elevating theta in the frontal cortex, SMR in the medial cortex, motor cortex, and they're decreasing theta in the frontal cortex, increasing alpha SMR in the central cortex, and suppressing alpha in the posterior cortex. Now, what state? Only one person got this in Denver. This person's falling asleep. The frontal system has gone into alpha, to theta. They're not moving. That means that they're not processing. They're not moving, so you have a great SMR. And alpha is suppressed, so they're paying attention. They're not. Well, that should have an alpha up. That was a bad choice. I'm sorry about that. This is someone who's actually doing a good job. The sleep is the next one. They're suppressing frontal theta. They're increasing SMR, and they're suppressing alpha. And so if you wanted to ask yourself, what kind of brain state would you like to have a subject achieve when you're trying to learn to control feedback displays? You want them processing the data, you want them not moving around, and you want them paying strict visual attention. And that's exactly what we have here. And then we have covariance measure, which will show you. Unfortunately, this got cut off a little bit. Let's go on to a good one. Okay, so this is the one that's sleep. I think it is. No, this is another good one. This is okay. So the earlier one of these is when we just ran this out. This is one of the things that we learned that we didn't expect. If you just record a baseline with no feedback, just a raw EEG in the context of training, if alpha goes up, if SMR goes up, theta and alpha always come down. If you then add training to it, you have theta and alpha both come down much more profoundly, and you have a bigger SMR. But it is natural, as we've seen from the other earlier information, that just producing SMR, which means quieting motor excitability, makes it impossible to have better attentional skill and be less fidgety, I suppose is a good word. Now, we got started on some patients with a problem. So this is an epileptic, and they were able to pull the theta down, but they had more theta than almost anybody. This took up a whole segment of the analysis. Now, here's the SMR, the covariance study. So you tell what's going up and what's going down. So the alpha goes down and SMR goes up. That's all that says. Here again, you have training going with the process instead of just baseline. And you're getting profound suppression of theta, marked increase of SMR, and they're supposed to all occur. The reward occurs right here, and marked suppression of alpha. Now this is a different patient. This is a highly anxious patient, and we could not turn her off. Okay, she is producing theta here, and is fidgety, cannot stabilize her motor excitation. She's in and out of whatever she's going through here. But she is able to pay strict attention to what she's trying to do, but she's getting frustrated. And her issue was anxiety. And we've seen that. Here's another one like that. So we're now starting, and then we can plot their achievements. So I'll just say what can we conclude now, because even 2 hours wasn't enough. There is valuable information provided by the spontaneous EEG if one knows what they are seeing. Spontaneous EEG rhythm patterns have been decoded, not only in terms of Greek names, but in terms of specific origins and the functions they reveal. These functional states and their interactions can be followed spontaneously in relation to neurofeedback training. Neurofeedback can thus be applied to correct both errors in the network regulation and in treatment strategy, period. Thank you for tolerating all of this. I'd ask you to rethink this. I don't care what I'm doing as long as it works. I don't care if it works if I don't know what I'm doing. You take your choice. Although if you take the wrong one, you end up looking like this. So, how are we doing with time, Andrew?
We've got about 4 minutes left. So let's see what we can do with 4 minutes.
All right. Well, I'm sure there are some questions, either that or they all have headaches. But I'll be happy to answer what I can. I'm sorry, I don't love this format, and there was a lot of material that I had to cover, but I hope you realize that this is all documented, reliable data, and the conclusions are based on facts, not opinion. Okay? So whatever you want to ask, please do.
Yes, and everyone still on the webinar, feel free to submit questions through the questions feature. And as you send those in, I will go ahead and try to read those off. Looks like someone tried to submit something but might have pushed enter too quickly. So we'll give it just another minute or two here and see if we can get that question submitted.
Let's see if Rusty is there. Rusty, are you there? No reply. It's a real challenge for me to present this. I'd like to make the whole talk about the last part, but I just won't let myself do it without having the evidence to support what I'm saying.
I think I just got something emailed to me. The question is, any connections between altering BF, thalamic activity, and the vagus system, particularly ventral vagus?
Yeah, actually, at one point we thought stimulating the cut uh, anterior end of the vagus was anticonvulsant, but that didn't hold up. I don't know. And there was, there have been various efforts to use vagal stimulation to alter brain excitability, but I don't know what the current literature is saying about that. So again, and what was the basic question again?
Sure. It was, are there any connections between altering VF, thalamic activity, and the vagus system, particularly the ventral vagus?
There probably has to be, because it does alter the autonomic nervous system in a variety of ways. So it would alter blood pressure. We weren't monitoring, the vagal system. There's so many things you could do. That's the problem. One of the things that pleased me when we finally began to see what was going on was that we could be fairly simple about it. I mean, it didn't take a lot of thought. You look at those three processes, cognitive engagement, motor engagement, and visual processing engagement, and you kind of had some idea of the state of the aneurysm. I've gotten to a point now where I can look at the data as it's coming out, or after we've collected it, and draw some very profound conclusions about what the brain was doing, what the subject was doing, and what I would do if I was going to train them, which is one of the questions that always bothered me. And I had it shown there where I showed the lady that was injured, had her frontal cortex damaged and had frontal fast activity. And we helped her dramatically with skill at that time. But I couldn't figure out why, she really got better. And now that I know that the SMR and basal forebrain produced rhythmic activity, everything that we do with SMR and basal forebrain is inhibitory. And that gives us the other side of the game that we need to have in studying what the nervous system is doing and what the signals are telling us. And I think that's very useful. And the key didn't help me because I could look back at that frontal activity that was fast and see what happened, but I didn't have that at that time. So this is just beginning. We're still collecting data. And again, if anybody's still there, anybody who's interested in joining us and lives close enough to do that, I'd be happy to talk to them.
All right, we do have a couple other questions, Dr. Sturman, but it is one past the hour. So I will go ahead and shoot those to you via email. And to the people who did submit questions that we didn't address, we will send those out. We will send those responses out to you after Dr. Sturman is able to address those. So again, thank you so much, Dr. Sturman, for spending some time with us this afternoon and covering quite a bit of territory with regards to your research and some of your new research as well. And thank you to all of you for attending today's webinar. I will be emailing you shortly an evaluation and the exam. Please complete this within 1 week, and you'll receive your 2 continuing education credits along with your certificate within a week after the deadline. Again, if you think of any other questions after we end the webinar today, feel free to email those directly to me or to Dr. Sturman, and we'll get those addressed. Thank you so much, and enjoy the rest of your day.
My email address?
I'm sorry?
Can you give them my email address?
I certainly will. Yep, I'll include that in my email.
Okay, because I'd be happy to. I have questions of them.
All right. Well, thank you, everyone, and we hope to have you on a future AAPB webinar. Thanks so much.